Hello. Prokaryotes and eukaryotes have diversified and have been independent for a while during the course of evolution. And therefore, there are also differences in the way the genetic information is expressed. And in terms of the mRNA, there are big differences, and that's what we'll try to have a look at it now. So the mRNA is the one that is produced after transcription from using the template DNA as the template. And the mRNAs that are produced in prokaryotes and eukaryotes have subtle differences, and that's what we will try to have a look at now. Say in the mRNAs, they can see the prokaryotes and eukaryotes. What we see here is that in prokaryotes, we see that there are 3 open reading frames. Say in this case, this is one reading frame because we have a start codon and a stop codon, and we have another start and another stop. And so this is a second open reading frame. Here is the third open reading frame, start and stop. So one mRNA can consist of more than one open reading frame. This is especially because in prokaryotes, the genes are organized. All those with related functions are usually put as one operon, which is there has a promoter, and then we have 3 independent open reading frames, and finally a terminator. So one mRNA is produced, and that is the polycistronic mRNA. And each of them have, although there are these open reading frames in the DNA or the genes are transcribed as one mRNA, each of these open reading frames have their own ribosome binding site. And so the translation, however, is therefore different in the sense that translation of the first open reading frame starts here and stops here, giving rise to one protein. Similarly, another ribosome binding site is there, which in prokaryotes is called as Shine-Dalgarno sequence or SD sequence. And the ribosome is organized here, the initiation complex, and then translation of the second open reading frame would give a different protein, and the third one would give a different protein. So the 5' end of the mRNA consists of 3 phosphates like this in the prokaryotic mRNA. That's because the first base that is added, the pyrophosphate, is not released. So we have these triphosphate in the 5' end. And the 3' end, there is not much of modifications whatsoever. Whereas in eukaryotes, we have differences. In the 5' we have a G cap, that is a guanidine added, guanylyl GTP is added, and therefore we have a G base with 3 phosphates and the start of the mRNA. And on the end, 3' end of it, we have poly A tail, that is about 200 adenine molecules added. And they are monocistronic in the sense that there is only one open reading frame. That's the most typical way. It starts here and ends here, giving rise to a protein. This is important to also to recollect these things, because it's the translation. When we study about translation, we study prokaryotes and eukaryotes in parallel. So it will be useful that way. The ribosome binding site, especially in the prokaryotes, binds is something like GGA-GG. That is called as the Shine-Dalgarno sequence is said. It has sequence or complementarity with the 16S rRNA that is present in the small ribosomal subunit. And that is how the ribosome recognizes the mRNA and then starts translation. Of course, the initiation complex, the large subunit binds to it, and then initiation happens. But there are mechanisms for the ribosome to recognize or identify which mRNAs should be, or which RNAs in general to be bound. Those are the mRNAs, and where to bind and from where to start translation and where to terminate. And everything is in the mRNA. Here we have a ribosome binding site, then we have start codon, that's where the amino acids start being added, and then here we'll have the termination. So everything is there in the information, in the mRNA, and all the ribosome and the rest of the translational machinery come into place and make this complex process happen. Thank you very much.